A team at Stanford University has demonstrated a new approach to reversing memory loss in mice. An infusion of spinal fluid from young mice reversed the memory loss typically seen in aging animals, the team reported this month in the journal Nature. A growth factor found in the fluid also improved memory, though to a lesser degree, says Tony Wyss-Coray, a neuroscientist and senior author of the study. When we put the factor in mice, they actually are better able to perform a memory task where they have to remember something that happened to. The finding hints at a whole new era in the search for treatments for Alzheimer's disease and other age-related conditions that affect a person's memory and thinking, says Maria Lehtinen, a neuroscientist at Harvard Medical School, who wrote a commentary that accompanied the study. So far, most efforts to treat Alzheimer's have focused on eliminating the disease's hallmarks, toxic plaques and tangles that build up in the brain. Those efforts have produced drugs that can reduce the plaques and tangles, but have yet to do much to preserve a person's thinking and memory. The latest results with spinal fluid suggest other treatments can help, even if they don't affect the underlying disease process. What we're seeing is that there's much more going on and that aging seems to produce a lot of abnormalities that contribute to cognitive decline and dementia, Wyss-Coray. The new study involved elderly mice, who, like their human counterparts, tend to develop memory problems. For example, a painful experience will form a memory that lasts for weeks or even months in a young animal, Wyss-Coray says. As a result, the animals will continue to freeze in response to a light or sound cue that was once accompanied by an electric shock. When they're old, they keep forgetting about this, he says. A few days later they can't remember that they were in a bad environment. Wyss-Coray's lab wondered whether one cause of their fading memory might involve cerebrospinal fluid, or CSF, which bathes the brain and spinal cord. Brain cells depend on this fluid, and its composition changes dramatically as an animal gets older. But they hadn't tested the idea because extracting spinal fluid from a tiny animal is so difficult. That changed, thanks to Tal Iram, a postdoctoral research fellow in the lab who was determined to overcome the technical obstacles.